All right, we can get started for today. So first, I'd like to welcome everyone for joining us for today's webinar. My name is Kyle Foster, and I am a content marketing scientist representing Biocytogen's marketing team in Boston, and I'll be moderating today's webinar. I'm joined today by our speaker, Dr. Frank Ahn, who is the Senior Director of Antibody Therapeutics at Biocytogen. His key responsibilities include overseeing Biocytogen's internal antibody discovery programs, including portfolio strategy, target selection, and project implementation and management. Dr. Ahn obtained his PhD in cellular and molecular biology at Brandeis University, followed by a postdoctoral fellowship in immunology at Dana-Farber Cancer Institute and Harvard Medical School. He has over 20 years of experience in drug discovery, both in industry and academic settings. Today, Dr. Ahn will be discussing the current landscape of bispecific ADCs as novel therapeutic modality, and we'll be providing an introduction to Biocytogen's bispecific ADC platform, highlighting the recent progress of several of our company's bispecific ADC assets. Before we dive into today's discussion, I would like to remind our viewers that the question and answer box is open. We will address as many questions as we can at the end of today's discussion. And with that, I'd like to invite Dr. Ahn to begin today's talk.
Thank you, Kyle, for the nice introduction. And welcome again for coming to the webinar. And today I'm going to talk about our Biocytogen's bispecific ADC programs. So here are today's topics. I'm going to give a brief overview of our company and our antibody discovery platform, including that for bispecific ADC. And then I'm going to focus most of the time on two case studies to illustrate how we make or design and generate bispecific ADCs at Biocytogen. So there are two stories. And then in between, I'm going to introduce our, new and differentiated topoisomerase 1 based payload and the linker system, then followed lastly by a conclusion remark. So first, let's take a look at Biocytogen and Biocytogen's antibody discovery platforms. So Biocytogen is a biotech company. We are a public company with over 1,000 employees, with headquarters in Beijing and a large animal facility in Jiangsu province. And we are international. We have a very active team in Boston performing pharmacology services to many biotech and pharma companies. We also have business offices in San Francisco in the US and also Heidelberg in Germany. So let me see, move this. We just want a brief introduction to what we have accomplished in the last about 3 years is that we actually generated the library of fully human antibodies that contains over 400,000 characterized fully human antibody sequences. So that's one dramatic achievement for Biocytogen. And then we also, have very active in the partnership side or licensing side, and we have struck 103 asset licensing agreements with farmers and biotechs alike. So that's, in a nutshell, is an overview of Biocytogen and the antibody discovery part. So just to give you a little bit more granularity, Biocytogen has two separate business arms. The first is called RenBiologics, and that engages in antibody discovery and co-development. And we also have a licensing arm that help the discovery team to license out the asset molecules. The second arm is called BioMass. It is the fee-for-service CRO-like service provider and that provides pharmacology services and the disease models. And we have the world-class gene editing team that can generate any genetic model, animal models for you. And we also have a large-scale animal facility, as I mentioned, in Jiangsu province, where we house about 800,000 mice every day. So that's kind of the two business arms, and we are going to focus on the Ren biologics for today's seminar. These are the modalities that bsADCs antibody discovery team have been working on. The first is a monoclonal antibody, and the second is bispecific antibody platform. Then derived from bispecific antibody platform is the bispecific ADC platform, which is today's topic. And then we'll also work on fully human single domain antibodies, usually commonly known as nanobodies. And we recently have a fully human T-cell receptor mice that can be used to discover the T-cell receptors that recognizes the T-cell receptors that recognize specific antigen and MHC complex. And so that's on the TCR side. And similarly, we have a TCR mimic platform that can be used to discover antibodies that recognize the peptide and MHC complex. And both platforms, by nature, they can recognize intracellular antigens, versus the other platform that would usually recognize extracellular antigens, meaning surface proteins, cell membrane, plasma membrane surface proteins, or secreted proteins. And we also have a team that work on the hard-to-target GPCR targets using the Ren mice KOs, using a special strain of mice where we knocked out some of the targets to expose more epitopes for antibodies. So all these are predicated on a suite of fully human transgenic mice we collectively call the Ren mice, and they have different versions like a RenMab that is useful for generating monoclonal antibodies, and the RenLite, which I will give a brief introduction in the following slides, that is very good for generating bispecific antibodies, and the RenNano mice for nanobodies, RenTCR mice for fully human TCRs, and etc. Okay, so this is kind of the engine for our antibody discovery. So everything coming out from bsADCs are fully human antibodies. But today we're going to focus on the bispecific ADC platform. So before I get into the bispecific antibodies and ADCs, I just would like to point out a very important component in our antibody discovery. That is codenamed Project Integrum, which is essentially an early stage discovery effort to generate antibody binders for over 1,000 targets. So this was motivated by the gap between the number, a small number of clinically validated targets that includes approved drugs, and also some drugs that are in clinical trials and their targets. That's a small number versus a much larger number, about 1,400 of them, potentially druggable extracellular targets. So our motivation was to fill this discovery pipeline, to fill this gap, between 1,400 and about 100 of them, so that more targets and drugs can enter this clinical pipeline. So as I mentioned, we have achieved a lot in the last few years. So we have done over 900 projects trying to discover antibody binders either in the RenLite or in the RenMab or RenNano format. And we, as I mentioned earlier, we have over 400,000 characterized fully human antibody sequences. And we also have done, biocytogen ADCs, biocytogen antibodies, and the TCR mimic antibodies. And here, I listed some of the targets that we have been working on. And here they are divided according to therapeutic areas, like in oncology and inflammation and autoimmune diseases, neuroscience, metabolism, and cardiovascular diseases. Okay, just to give you a flavor of what we have been working on. And this is, as I mentioned, we have done over 100 antibody assets partnerships with pharma companies and also with biotechs. Okay, so that's about Project Integrin. It is important because a lot of the tumor-associated antigens or TAAs that we use for bispecific ADCs came from this initiative. Okay, now let's delve into how we generate the bispecific ADCs. And the first is the bispecific target recognition modality, the antibody part. So as I mentioned, we use the RenLite mouse to generate the bispecific antibodies, and these mice are common light chain, and so the antibodies share the same light chain for the two arms, and they were designed specifically for making bispecific antibodies. So this shows how we constructed the mouse strain. So on the left is the heavy chain locus. Here's the top is the mouse heavy chain locus. Essentially, we replaced this heavy chain immunoglobulin gene locus in one step with the human heavy chain locus. So in this way, the resulting mouse will have only the human heavy chain, but it has the entire repertoire of the human heavy chain genes. So that's on the heavy chain side. On the light chain side, we replaced the mouse kappa locus, okay, the entire locus, with a single human kappa gene, 3-11. So in this mouse, all the heavy chains can only pair with this single light chain, so making this light chain a common one for all the heavy chains. So in this mouse, all the antibodies uses the same light chain to make functional antibodies. So we have done a lot of characterizations of this mouse strain called RenLite, and we know that they can generate very robust immune responses comparable to wild-type mice, and we can generate very diverse heavy chain repertoire and similar to that of the wild-type mice against any targets. And these RenLite clones or antibodies, they have very good affinity up to low to sub-nanomolar KDs, and they carry very few somatic mutations on a common light chain, so making them a bona fide same light chain between different clones, and that making them a very easy very convenient in constructing bispecific or multispecific antibodies. This is because when you have a common light chain, like in this case of two different antibodies that targets two different antigens, and all you need to do is express the heavy chain, two different heavy chains, and this common light chain, 3 molecules in the same cell, and then they will assemble into the right configuration of functional antibodies because there's no issue between the correct pairing of light chain and heavy chain. So this is in contrast to if you have two different light chains and two different heavy chains. When you express them in the same system, only a fraction of the resulting antibodies will be the correct ones. Of course, in both cases, we use the knobs into holes technology to pair the heavy chains correctly. Okay, so that's the biggest advantage of common light chains versus just the two regular antibodies in making the bispecific antibodies. So now turn to the ADC. Why do we want to do bispecific ADCs versus a single targeting ADC that has been known for years in our industry? So we believe there are a few advantages of bispecific ADCs over single targeting ADCs as follows. So the first is improved efficacy, because bispecific ADCs by definition recognizes have two arms and each arm recognizing one epitope. Okay, so this antibody recognizes two different epitopes. And by carefully selecting the two arms can have synergy in internalization and cytotoxicity. That you are going to see some data that supports this. So the first advantage is the improved efficacy. Then the next advantage is that bispecific ADCs helps overcome tumor heterogeneity and reducing resistance. This is because you would hit the tumor with an antibody that hit two targets at the same time. So that helps cut down the potential resistance from the tumor. And lastly, the bispecific ADCs also potentially can help increase tumor binding specificity, because they have two arms. By fine-tuning the two arms, you can have the tumor, you can have the antibody bind to the tumor that expresses both antigens better. And so this would help reduce the off-tumor on-target side effect in normal cells. So these seems to have been borne out by recent clinical trial data. Here I just list, here is a HER3 targeted ADC, patritumab DXd. So that's a single targeting ADC. And on the right is a Bilee's molecule that targets EGFR and HER3. Okay, so here is the response in the phase 1 trial. And this targets EGFR mutant non-small cell lung cancer, or EGFR wild type non-small cell lung cancer, and EGFR mutant non-small cell lung cancer. The same for Bilee's bispecific ADC here, and they share the same class of payload. So we just consider they are very similar, but then you look at the efficacy side, you can see either for the wild type EGFR or the mutant EGFR, the bispecific ADCs outperformed the single targeting HER3 ADC. So suggesting that by targeting two targets, the bispecific ADCs worked more effectively. Of course, these are cross-trial data for comparison, so we have to take it with a grain of salt. But nevertheless, that there's excitement in the field that bispecific ADCs could do a better job than single targeting ADCs. And in the field started to really expand in the last year to a year and a half. when they just started with only one bispecific ADCs in clinic to about a year and a half ago to the end of 2020, there's one more addition from AstraZeneca that targets EGFR and the CMET to, after Bali reported their molecule, the clinical trial results in last year's ASCO, now in this year's AACR, that was just, a couple of weeks ago, and there are many, many posters, abstracts talking about bispecific ADCs. And so the field is really growing as we speak. So with this introduction, I'm going to turn to some specific cases of how we generate ADCs and how the ADCs perform in our in vitro and in vivo studies. I think that would be the best way to illustrate how these ADCs could potentially be effective molecules helping patients fighting against cancer. So the first case study is a molecule codenamed YH12 that targets TROP2 and HER2. So it's a bispecific ADC. So this slide shows you the structure of YH12. This is a common light chain indicated by the green color RenLite antibody that targets HER2 and TROP2 and conjugated to the classic VC MMAE linker and the payload with a DAR of 4. And in addition to this bispecific ADC, in our studies, we usually also make these control antibodies that are the parental monoclonal anti-HER2 and anti-TROP2 ADCs or antibodies, and also the monovalent, one arm, and monovalent HER2 or monovalent TROP2 antibodies, because we wanted to compare all the activities of all these antibodies to demonstrate the advantages of the bispecific ADCs. So the selection of these two targets were not random, and as we all know, HER2 and the TROP2, the ADCs, they are approved drugs for breast cancer. So this is the in HER2 Destiny Breast 04 data, clinical trials results, part of the results. So during the trial, this trial showed that in HER2 actually provided a great overall survival benefit to patients. However, if you look at the response data, here's from the Breast 04 trial, the response rate, ORR, is only just about 50%. So that indicates about half of the patients actually did not reach the response that can be characterized as a responder. So that our hope was that by targeting both HER2 and the TROP2, we can push this bar even higher so that more patients could benefit from this therapy. Okay, so now I'm going to talk about a series of data of YH12 that actually demonstrates the advantages of using a bispecific antibody or targeting a dual targeting bispecific ADC advantages over the single targeting ADCs. So the first is the binding of naked antibodies, and we have two cell lines here. That one cell line expresses high level of HER2 and high level of TROP2, and you can see for the YH12, which is this orange line, this bispecific molecule binds much better than the two single targeting antibodies. the parental anti-HER2 antibodies and the parental anti-TROP2 antibodies here. The similar pattern for the MCF7 cell line that has a low HER2 expression, so the HER2, the green line was lower, and intermediate the TROP2 expression, so which is indicated by the blue line, but the orange line that represents YH12, the bispecific ADC was still the best one. So that's binding. And here is the internalization data on NCI N87. That is the first cell line that I showed the binding data of. And so we use the InQSite to, this is a live monitoring of the internalized signal, which is a pH-sensitive dye. So when it reaches the lysosome, the pH changes and it emits fluorescence. So this red line is the internalization signal activity by YH12. And you can see it's very robust internalization. This is in contrast to the two parental anti-HER2 and anti-TROP2 antibodies indicated by these bluish lines, and also much better than the monovalent parental HER2 TROP2 antibodies indicated by these two green lines here. Okay, so if you tabulated the data, let's say we take a look at it after 24 hours, and here is the result, and you can see the red bar, that is the bispecific activity, is a lot more than the sum of the two green bars that represent the monovalent anti-HER2 and anti-TROP2 antibodies. So clearly there's some synergy going on between the two arms that would make the bispecific ADCs internalizes much better. This is the in vitro cytotoxicity data. And again, here YH12 is represented by the red curve here. And the two green curves are the monovalent parental antibodies. And you can see, again, the bispecific killed the N87 cells in vitro in a much effective manner. And this is the CDX xenograft in vivo data. And actually, there's a few things that I would like to explain to you. This is an interesting data set. So first, you're going to see there's a clear dose response of YH12 indicated by the different shades of the orange colored lines here. And then the second is that, just as I just showed you in vitro, that if you look at the, let's see, here's 1 mg per kg dose, okay, and compared to these two green lines, okay, the bispecific that represents the parental HER2 and the TROP2 ADCs, here everything is ADC. And so the darker orange line outperformed the two parental antibodies, which were the two green lines here. Okay, so that's second point. And then the third point is that if you look at the YH12 at 1 mg per kg, which is this line, and you compare a combination of the same number of paratopes. Okay, here the parental HER2, half of the dose because it has two arms, so it's half of the dose. In combination with a TROP2 ADC, okay, also half the dose of the bispecifics. So they have the same number of paratopes inside the mice. However, the activity, okay, when you put these two paratopes together on the same molecule, the antibody or the bispecific ADC worked much more efficiently than the combination of these two epitopes on separate molecules. So that is a very good piece of evidence that supports our claim that the bispecific ADCs can work more efficaciously than the parental antibodies separately. So that's a xenograft data. And now I'm going to show you the PDX model that has a low HER2 expression because, as we all know, that it is the HER2 low population of breast cancer patients that we are most interested in because they are the largest population for in breast cancer patients. So here again, you will have two different doses, 6 mg per kg, and YH12 indicated, and 3 mg per kg indicated by the two different shades of the orange lines here. And then this is compared to the two parental ADCs, anti-HER2, anti-TROP2, the two green lines, the monovalent anti-HER2 and anti-TROP2 ADCs represented by the purplish lines here. So as you can see, the 3 mg per kg, which is this, that's the bispecific ADC activity, this outperformed both the parental monoclonal ADCs or the monovalent parental ADCs. So that is about the comparisons of different ADC molecules to show the outperformance of the bispecific molecule. And here we compared the bispecific ADCs to a few approved drugs that targets TROP2, and these two targets HER2. And you can see in this particular model that our bispecific ADCs work better than the approved drugs that targets in a singular manner of either TROP2 or HER2. So basically what I showed you was with the in vivo data is that the bispecific ADCs performed better than the parental ADCs, either in the bivalent form or the monovalent form, and there was synergy between the two arms. So this phenomenon actually was not unique to YH12. So I'm going to quickly show you two other bispecific ADC molecules. The first one is a TROP2 and the PTK7 bispecific ADC. We all know what the TROP2 is, and the PTK7 is a pseudo transmembrane tyrosine kinase. So it's inactive, but it's overexpressed in a number of cancers, that including the ovarian cancer, non-small cell lung cancer, and triple negative breast cancer. So, and this is Pfizer AbbVie's molecule, and in the phase 1 trial, it shows as a single targeting ADC that showed welcome activity in ovarian cancer and also the triple negative breast cancer in the 20s of ORR. So, so we constructed this bispecific antibody and conjugated to vcMMAE. And here I just focus on this panel. And I think the PDX is balanced in terms of expression of PTK7 and the TROP2. Trying to move these things, and the TROP2, and so here everything is 5 mg per kg. For all these ADCs, except there was this higher dose of this is BCG, codenamed BCG 033. So there's a higher dose of BCG 33 here. That it was most potent, but here what I also wanted to point out is that the lower dose, 5 mg per kg, the bispecific ADCs also outperformed the two parental monoclonal ADCs, the individual ones, that one against PTK7 alone and one against TROP2 alone. So that I showed you that, this is what we observed again and again. The bispecific ADCs worked better than the single targeting parental ADCs. This is another molecule we codenamed BCG19 that targets EGFR and HER3. And it uses a different payload, which I'm going to talk to you about, but ignore that. And here you can see that here the red line represents the BCG19 that targets EGFR and HER3. And so as with YH12, this one worked better than the green line here. That is the combo of the two individual single targeting parental ADCs here. So again, you see that when you put the same number of paratopes on one molecule, it works more efficiently. So I just wanted to show you there's a certain general, you can generalize that to a certain degree that we can observe the bispecific ADCs work better than their parental individual parental ADCs here. So back to YH12, this molecule has been licensed, and so it's going through the CMC stage as we speak. So YH12 and most of the data I showed you were based on the vcMMAE linker and the payload. So since then, we have evolved into a new linker and a payload, as many in the field, that is DNA topoisomerase 1 inhibitor based and codenamed BLD1102. So before I introduce and describe some of our more recent data of bispecific ADCs, I would like to briefly introduce what are some of the properties of this linker and the payload system. So here is a summary of BLD1102. And as I mentioned, it's a DNA topoisomerase 1 inhibitor, and it has a potency just slightly better than the DXD, which is used in HER2. And it has also a moderately more potent bystander killing effect than DXD, and it can have a DAR of 8 via cysteine conjugation. So this linker is cleavable, and but one unique feature of that is highly hydrophilic. Okay, I'm going to show some data about that, and also has a very good plasma stability. Here is the IC50 data of the free payload part. It's called the BCPT02. So here you can see the IC50 of our payload was better, was better than the DXD. And this data shows that our payload did what we expected it would do. So these are the DNA damage markers and the caspase marker, and you can see after a few days treatment, these markers were upregulated, indicating that our payload worked with the mechanism that we expected. This is the bystander effect mixed culture assay, where on the left you have a double positive cell line A431, and on the right the two peaks is the double negative cell line H520. So here just focus on the top panels, and in the cold culture situation, if you treat it with isotype control, the cell numbers did not change. And however, if you use a ADC that recognizes both targets, both the, I recognize the two antigens on the surface, and you can see both peaks almost disappeared. Okay, and it's not surprising that the double positive peaks disappeared, but the double negative peaks also almost disappeared, and by definition, that was due to the bystander killing. This is in contrast to the DXD payload. Here, you can see there were still some more cells remained. And if you plotted that, and you look on, let's only focus on the black bar here, that represents the double negative cells or the bystander killing indicator. And you can see essentially the signals changed from this level to this level for our BLD 1102 linker and the payload, and versus the DXD linker and the payload here. So ours was a little bit more potent in terms of bystander killing. This is the hydrophilicity indicator for our payload. Here is that here is the conjugated ADC here versus the naked antibody. Here in this case is the trastuzumab. And you can see that when you conjugate it with BLD 1102, essentially it migrates to the naked antibody position, indicating that this is the Hick column, indicating that the linker and the payloads were very hydrophilic. This is in contrast to DXD, that you clearly see a rightward shift that indicates much higher hydrophobicity. So the similar result we got from the reverse phase HPLC that measures the hydrophilicity here. And here the case is star of 8. Here's the plasma stability data of BLD 1102, and with human, monkey, rat, and mouse plasma. And the highest degradation was observed after 14 days was in human plasma, which is under 2%. And to put this in perspective, in HER2, the DXD linker and the payload, I think for human it's just slightly above 2%. But in monkey, that can be as high as 4%, close to 4% degradation after 14 days. So this data just showed that at least our linker and the payload is as stable, or maybe a little bit more stable than that of HER2. So we have confidence that once it's got into clinical trials, this linker and the payload will perform in the stable manner. This is just very quickly, this is in vivo data that we wanted to show that our linker and the payload was more effective or efficacious than the DXD linker and the payload. So here is just the orange line is our payload, and then the blue line indicate the DXD linker and the payload. That's one PDX. Here's another PDX, the two different doses, showing the same conclusion. So with that introduction of the linker, I'm going to show you actually how it performed in another Bispecific ADC that targets EGFR and the TROP2. We codename that DM001. This is what's actually licensed to another biotech company called DOMA. So before I go into the new linker and the payload, we actually performed a lot of assays in vitro and in vivo with VC-MMAE payload on this bispecific molecule, again from the RenLite platform. So without going through the details, I just summarized the activities here that we observed the DM001 Bispecific antibodies internalized much better than the parental monovalent antibodies. and the bispecific ADCs had stronger cytotoxicity in vitro, and it potently inhibited, CDX and PDX models in vivo. And in the PDX, we also showed that with the MMAE linker and the payload, the bispecific ADCs worked better than the parental, and then the benchmark ADCs, and also the parental single targeting ADCs. So that is just how the antibodies performed with the naked antibodies of the vcMMAE ADCs. Here are the data with the new linker and the payload, which is the DM001 with the DAR of 8. So again, here is the CDX model, and you see a clear dose response curves here. And if you look at the higher dose, that is this DM001 activity, and it was performed better than the two parental anti-TROP2 or anti-EGFR ADCs here. And here we again compared, different doses, and also compared it to the known anti-TROP2 drug, sacituzumab govitecan. And even when they have to dose, and also cetuximab, and even they dose these two controls, approved drugs, much more frequently than the DM001. And the DM001 at the two different doses still outperformed these approved drugs. I have to say that the cetuximab is not a really apple-to-apple comparison, because this is just an antibody, it's not an ADC. But nevertheless, it is approved EGFR antibodies, so we included that as a control. The two data sets shows that DM001 at the two different doses and worked better than the vcMMAE linker and the payload. So here you see that there's two different shades to indicate two different doses, and you can see at the lower dose that the new linker BLD1102 outperformed vcMMAE, although at the higher dose they performed very similarly and almost completely inhibited the growth of this PDX. But in the different PDX, and you can see there's a much larger difference where both doses of DM001, which means with the new linker and the payload, outperformed the vcMMAE linker and the payload here. Then we performed a series of PDX studies with DM001 that include many different types of cancer or PDX models, from like a triple negative breast cancer, esophageal cancer, and different mutations of non-small cell lung cancer, or the wild type non-small cell lung cancer PDXs, and head and neck CRCs, and gastric cancer, and here is the waterfall plot of the best percentage change of these PDX tumor volumes from baseline. And so you can see that there's some tumors were more sensitive to DM001 that has had the new linker and the payload, and some were more resistant. And there's no straightforward correlation between the expression levels versus the efficacy, because you have on one end you have this here's the H-scores for both in the mid 200, and you have about 200 on the other end that was less sensitive So we don't observe any kind of a expression level dependent um, uh, sensitivity. However, we could not help but recognizing that it might have a cancer type sensitivity. For example, here is that the two best uh, uh, the, the two best performing PDXs were from gastric cancer. So they have 3 gastric cancers. They were all responsive, much more versus the CRC PDXs that tend to be less sensitive to DM001. So with that, we also performed a pilot DM001 NHP assay. This was for the dose range finding components of the safety assessment on cyno monkeys. And when we increase the dose from 5 mg per kg to 20 mg per kg every 3 weeks, and so we actually did not observe general toxicities, all visible lesions in the tissue of interest that includes the skin or lung. And we did observe some white blood cells and immature red blood cells counts decrease, but they were transient and restored over time. So overall, it appears that this bispecific antibody was reasonably safe in monkeys. So the real safety assessment has been ongoing. And so the next app that I'm going to would like to show you is actually the developability of this molecule. And here we subjected the molecule to different stress conditions, like raise the temperature to 40 degrees for 7 days, or subjected the molecule to a low pH environment, and we use different analytical methods to track the integrity of this molecule. And as you can see, in all cases, that the molecule remained stable and using different analytical means. So that was good. And here is our, cell line development data. And here the cell line grew really well. And here I just wanted to point out that the titers can be very good, 6+ grams per liter. And the purity was also very good, almost 100% pure after just a straightforward purification after the affinity purification step. So this just want to remind people that this was from our RenLite common light chain platform. So this is a common light chain bispecific antibody, and at the CMC stage, it provided a great titer and a great purity. So we have a number of over 20 bispecific ADC programs in-house that are currently ongoing. And we also have about 200 or slightly over 200 TAAs that are, that can be used as like a plug and play. So in that, if someone knew the biologies of 2 different targets and they wanted to make a bispecific molecule, they can just, check our library. And if we have the clones, then you can, here are the targets that we do have clones, and you can make a bispecific antibody very conveniently using our common light chain technology. So here are just listed 7 abstracts that we presented at this year's AACR. And in our website, I think you can download them if you are interested in any of them. And to summarize my talk, about biocytogen, we work on therapeutic antibody discovery, and then mice, the fully human transgenic mice, a suite of them are the foundations and engine for our antibody discovery. So for our ADC programs, we focused on bsADCs because they have a number of advantages over single targeting ADCs, and we have a library of about 200 targeting. We have a library of binders that targets 200 TAA targets, many of them through Project Integrum. And these antibodies are common light chain format that is very convenient to accurately assemble into bispecific targeting antibodies. And we also have a novel and differentiated and functionally improved linker and payload for our more recent ADCs. So the lead molecules based on our bsADC strategy are right now planned to enter clinics later this year and early next year. And we are also very open to licensing and collaboration opportunities for these bsADCs at all stages, including the naked binders for the 200 TAA targets. So with that, that's reached the end of my talk. And I just wanted to thank the project teams for the nice data that I presented to you today. And thank you very much for your attention. If there's still time, and I would be happy to answer any questions. Thank you.
Thank you, Frank. We do have a couple of questions that have come in, so let's try our best to get to a couple of them. First one, how is the payload and linker selection made for the YH012 asset?
Okay. The YH12 uses the classical bsAMMAE, and that was one of our earliest molecules that we bsADCs we constructed, and around that time we only had actually bsAMMAE. But the licensed molecule was actually still carries the bsAMMAE that was we didn't convert that to the topoisomerase linker and payload. I didn't know exactly how the licensee company thought about that, but one complaint you heard if you went to those different conferences, ADC conferences, it was that people complained that too many companies are right now working on topoisomerase 1 inhibitor-based ADCs. So they were concerned about resistance. Okay, not too many ADCs were using the same class of payload. So maybe, the licensee companies was very smart that by choosing a bsAMMAE payload for breast cancer, they might actually position themselves smartly and in a nice way. And after HER2, which is topoisomerase 1 inhibitor-based linker and payload, I don't know if that's exactly the case, but I would choose that. I would choose that strategy by sticking to a different orthogonal linker and payload for this bispecific ADC. Hopefully, I explained that question, but if I was not being clear, please feel free to ask more questions.
Another question that came in a bit more broad. So are there, or is there the potential for bispecific ADCs to target, other diseases like diabetes or other metabolic syndromes, outside the realm of oncology?
Clearly, the people are thinking about the application of ADCs in general in other areas. Autoimmune diseases is one that people are actively trying to come up ways, both in terms of targeting and also in terms of the linker and the payload to be used for these. I know for sure people are working very actively and hard in those areas. Just I don't have a concrete example, a very successful example to show you, but I can assure you that the people are working in different areas other than oncology with either the single targeting ADCs like, MOA or bispecifics.
And I guess we'll just do one more due to time. How can one know the safety profile of bispecific ADCs before like the primate data is available, for example? Are there ways to predict that?
Huh. It is very challenging to know how you bispecific ADCs or actually any ADCs would be safe before you let's say before you in the preclinical stage and or before non-clinical studies. This is because the mouse or rat are just not a very good system for ADC for assessing the safety aspect of ADC because one key cytotoxicity actually came from the stability of the linker, and as we know that the different serum or blood will have a different impact on the stability of the linker and payload, particularly the linker, the stability of the linker. So people usually go to monkeys for safety assessment of ADCs, but before that, you could do something. Even though you just have to be conscious that they are in the rodent. But you could do like general tox studies to monitor the body weight. If you saw there's a clear sign of body weight increases at certain doses, that could raise a red flag or a yellow flag to alert you. You could use some humanized mouse strains to test body weight, to monitor body weight, etc. But in most cases, if you want to get a really reliable safety assessment, you have to go to monkey in non-clinical studies.
Okay, understood. Well, unfortunately, we don't have time for any more questions. I did want to thank you, Dr. An, for presenting today and for everyone online who tuned in for the discussion. This webinar will be available to watch on demand if you did miss anything during today's stream. Also, please don't hesitate to visit our website to learn more about our products and services. As Frank had mentioned before, we do have a lot of posters that highlight the assets that were discussed during today's talk. So feel free to check those out as well as download them. And also reaching out to our business development team through our website if you have any additional questions about anything that was discussed today, or if you have any inquiries about our antibody discovery platforms or assets. So with that, enjoy the rest of your day, everyone.